Good morning. Thank you very much, Marisa. Thank you a lot. I'd like to thank the organizers, mainly Mario Del Popolo, who invited me. It's a great pleasure and honor to give the opening lecture at this meeting with such a prestigious audience. Sorry, I have a bad throat. And it's always very nice to come back to Mendoza. So although my surname is a little bit funny, I was born and live in Buenos Aires. I'm not from Europe, but my ancestors were from Europe. So in the next 45 minutes, I have them because we are more than half an hour late, no problem. I'll try to share with you all results, some very new unpublished results on the epigenetic control of alternative splicing, and also how this knowledge could be applied to cure a disease. So alternative splicing is more a rule than an exception. It's estimated to affect the expression of 95% of the human genes. It explains how a vast protein diversity can be achieved with a limited number of genes. We don't have many more genes than worms or flies, but we make many more proteins, and that's because every gene of our genes can make more proteins than the flies or worms can make. Mutations that affect alternative splicing sequences that are called splicing enhancers and silencers are a source of human disease. Many hereditary diseases and cancers can be explained by mutations in splicing sequences. And the fourth issue has been the subject of my lab for the last 25 years, is that alternative splicing regulation not only depends on the interaction of splicing factors with their target sequences in the pre-messenger RNA, but is coupled to RNA polymerase II transcription. So this is the way I try to explain alternative splicing to lay people. I try to show them that the gene is the DNA is transcribed, and it makes a very long piece of fabric. It's called pre-mRNA. And that this fabric, many pieces of the same fabric, can be cut and sewed in different ways by the spliceosome to make different messenger RNA and make different proteins. And in this case, for instance, the same piece of fabric can be used to make a single-breasted jacket or double-breasted jacket, which are two different versions of the same issue, the same protein. So when I was in Oxford, as Marisa mentioned, we cloned the human fibronectin gene with Tito Barale, and we discovered its alternative splicing, and after different times and work, we found that there are 3 regions of splicing that could give rise to 20 different messengers and 20 different proteins from a single human gene. And at that time, we thought that was an extreme case, until we learned that there are even more extreme cases, like the case of the DSCAM gene in Drosophila, that can make up to 38,000 mRNA variants. And this is because it has 4 regions of alternative splicing with 12, 48, 33, and 2 alternative exons each, which are mutually exclusive. And this is not just noise. This is a noisy mechanism that allows for selection. I will not have time to explain these, but I will just mention the philosophical problem in the definition of a gene. This gene can make more proteins than the number of genes the fly has. So the fly has 18,000 genes, and now we have only one of them that is able to make 38,000 different proteins. And it has a function in the development of the nervous system, which I said I don't have time to show. So just to illustrate the importance of alternative splicing, I will show one example that for me is one of the most spectacular ones. And this is the case of the vampire bats. These bats contain in their face special structures, I know the zoologists know this, that are able to sense heat. And because they are blood feeders, they sense heat from the prey, and they can go and attack them and suck their blood. But the fact is that for the heat sensing, they have a special channel in the trigeminal ganglia that opens in the neurons at temperatures above 30 degrees, which allows for infrared sensing, and it's good for feeding. But this channel is not an invention of these bats. The channel is the same channel that exists in the rest of the neurons in the body, in the dorsal root ganglia, but the other channels open at temperatures above 43 degrees. And they are important for prevent noxious heat, because they make the hand to be withdrawn when you have fire. So the fact is that the difference between the two channels that come from the same gene is only a segment in the carboxy terminal domain that is present in the channel that opens at 43 degrees and is absent from the channel that opens at 30 degrees, which means that, and this difference is a difference in alternative splicing. So in evolution, alternative splicing allows for this adaptation without inventing a new gene, but having an already known gene to make the two variants. So we know that splicing is mostly co-transcriptional, and it happens while the mRNA is being made by RNA polymerase II. I will not show the evidence for this, but there's plenty of papers showing that splicing mainly occurs co-transcriptionally. But we learned from our lab in the late 90s that splicing and alternative splicing are coupled to RNA polymerase II transcription. And coupling is not only co-transcription. In co-transcription, you have a concurrent reaction. The splicing that excises the intron and ligases the exons can occur before the enzyme RNA polymerase II reaches the end of the gene. But when we call about coupling, we are talking about the influence of the transcription machinery or kinetics on the splicing reaction, and vice versa. So this is a coupled mechanism. And the modes of coupling are essentially two that are not mutually exclusive, and it could be changes in Pol-II elongation rate, what we call kinetic coupling, or changes in the recruitment of processing factors to the RNA polymerase II itself, to its carboxy terminal domain, to the chromatin or the nascent RNA, which we call recruitment coupling. So we will probably focus during my talk on the changes in Pol-II elongation that has been also the subject from my lab for the last maybe 15, 20 years. So I will talk about the role of transcription elongation, the effects of chromatin structure, and how this knowledge can be applied to try to cure spinal muscular atrophy. So we found many years ago that slow elongation could lead to higher exon inclusion. And the model that we prove in different ways is that if we have a gene with an alternative exon that has a weak 3' splice site that is competing with a strong 3' splice site, if Pol-II goes fast, then the two sites are presented simultaneously to the splicing machinery, and that would lead to skipping. But if Pol-II goes slow, there is time for recruitment to the weaker side before the stronger side is made, and that would lead to exon inclusion, because both sites are occupied by the splicing factors that will excite the introns and ligate the exons. But the fact is, in the model gene we were studying, the second intron is spliced first, and the first intron is spliced second. So it's not a problem of order of intron removal, it's a problem of recruitment of splicing factors, a window of opportunity while Pol-II is transcribing. So, as I said, slow elongation promotes higher exon inclusion, but genome-wide studies from our lab and many other labs show that 80% of the elongation-sensitive alternative splicing events respond to this rule. But there is a 20% in which slow elongation has the opposite effect. It promotes higher exon skipping. And how is that in the 20% of the cases? We found it with Gwendal Dujardin, a French postdoc in my lab in Buenos Aires. He stayed 3 years, and he found the mechanism by which when you have a particular alternative exon, if Pol-II goes normal, there is an inhibitor, in this case ETR3, that is not able to bind to its target site in the intron. So this would lead to inclusion. But if Pol-II goes low, then the inhibitor has more time to find its target site, then displace the positive factors and promote exon skipping. So this is exactly the opposite effect, and I want you to remind this case, because in the case of spinal muscular atrophy, this would be the case. We will have a case of an exon in which slow elongation will promote skipping. So the role of Pol-II elongation was confirmed by the use of a slow mutant of RNA polymerase II, and this is a work we've done many years ago together with David Bentley from Denver in the USA, and he created a mutation in the large subunit of RNA polymerase II, which is an arginine for a histidine, that causes the enzyme to be slower, to be not able to overcome poses in the chromatin. So we use a system developed by Jeffrey Corden in Johns Hopkins University, which is to transfect cells in culture with a mini gene which has an alternative exon, and then co-transfect the cells with plasmids expressing either the wild type or the slow mutant of the RNA polymerase. But in both cases, these two proteins contain a second mutation that makes the enzyme resistant to the antibiotic alpha-mannitine. So if we treat the cells with alpha-mannitine, the endogenous POL2 is killed, and all the DNA, the plasmid or the endogenous DNA, is transcribed by the resistant to alpha-mannitine POL2, which could be either wild type or slow. So essentially, we saw that for those cases like the fibronectin exon 33 or ED1, in which slow elongation caused higher inclusion, the level of inclusion increased by 4-fold when transcription was carried out by the slow polymerase. While in the other kind of the 20% set of exons that had the opposite effect, we see exactly which was expected. With slow elongation, we have higher exon skipping. So what can change POL2 elongation in a more physiological way? I need water. Because that mutant of POL2 doesn't exist in humans, doesn't exist in animals, and there is a paper published by my friend Javier Cáceres from Edinburgh that will appear in EMBO Journal in the few next weeks that has been accepted, in which he created the mutation in mice, and this is embryonic lethal. So if the speed is changed, the embryo doesn't progress. Although the embryonic cells can be grown in culture, and it's clear that there are a huge of changes in transcription and alternative splicing because of the slow polymerase. Sorry. So what can affect elongation not being that mutation that exists in Drosophila but not in humans? Well, essentially, it could be modulation of POL2 intrinsic activity by phosphorylations or association to elongation factors. And I will not talk about this. I just will show that we, in the lab, Manuel Muñoz, we published a Cell paper in 2009 and Cell reports in 2017, studied these in response to, for instance, DNA damage. UV light causes changes in elongation. But the theme that interests us in this talk is changes in the template chromatin structure that could limit or facilitate elongation. And we had a hint about this, that chromatin would affect alternative splicing regulation also many years ago with Sebastian Kadener, in which he showed that if he had a template DNA for splicing with an open chromatin state, then POL2 will go in fast, and we had a certain alternative splicing ratio. But if we allow that DNA to accommodate a more compact chromatin structure, then POL2 was going slower, and then you get more exon inclusion of the case we were studying. But this higher exon inclusion caused by chromatin compaction was reverted by treating the cells with trichostatin A, which is a histone deacetylase inhibitor, so it promotes histone acetylation, it opens the chromatin and allows for faster elongation. So you had the opposite effect. But at that time, we didn't know anything about histone marks or epigenetics, and we now know many of the meanings of certain post-translational modifications in histones, but I will just focus on histone A3 lysine 9 methylation, which is a repressive mark. This mark creates compact chromatic structure and inhibits elongation and transcription. And the opposite mark is the K9 acetylation that is a permissive mark which opens the chromatin and allows for more elongation and transcription. We might also refer to K27 methylation, that as K9 methylation is a repressive mark. So I will not have time to tell you all the evidence about our findings that chromatin marks will affect alternative splicing decisions. But essentially, the model is what we call alternative chromatin alternative splicing. And we can have a single gene, like the NCAM gene, with two different chromatin status. When we have neuron depolarization, the gene is opening the chromatin via K9 acetylation. And we are talking now K9 acetylation not at the promoter region, but intragenically in the gene body. So around the alternative splicing event, where Pol2 can go, in this case, faster. So in the case of the NCAM gene, this would lead to exon 18 exclusion. But the same gene, in a different situation during neuron differentiation, creates a more compact chromatin structure via K9 dimethylation and trimethylation, and this would go with slower Pol2 elongation and higher exon inclusion. So far is our basic knowledge about how different marks and how elongation can affect alternative splicing decisions. But the problem is, can this be used or applied in a disease to see changes in splicing that can be controlled by controlling chromatin statuses? Okay, so we decided to enter the field of spinal muscular atrophy. And since I was going fast, I will tell you why we entered this field. For years, I was reluctant to work in something that was applied, and I didn't have a motive to work on it. But then, one day, the families of the SMA, spinal muscular atrophy disease in Argentina, came to visit me. They knew I was working in splicing, and they knew that one of the promising cures for the disease was by regulating splicing. And they knew that because we have a colleague, I will present him to you, Adrian Krainer, who works at Cold Spring Harbor in New York, who was studying the way of controlling this disease by controlling splicing. So they said, if Adrian works in the States, we want you to work here in Argentina. And I said, well, but I don't have anything to offer you. And they say, we don't mind. They are so intelligent, they are more intelligent than the science authorities, because they say, we don't want you to cure the disease. We want you to work in Argentina in the disease, because there we have an idea what is going on outside. We'll have better tools, and you will instruct us, and we'll clarify what is good, what is bad. And we don't mind that you have results tomorrow. We want you to work here. We think you have. And they started to support me. Okay? So I started to see what I could do. This disease is terrible. In this disease, what happens is there is the absence of a protein that affects mainly, but not exclusively, motor neurons. And the motor neurons are wasted, and they cannot innervate the skeletal muscle. So the kids affected by this disease, if they are not ventilated, they will die in a few weeks after birth, depending on the degree of the disease. But the fact is that the protein that is not made in the patients is the protein SMN. And this SMN protein is part of the splicing machinery. So it's like a cycle. Okay? There's a protein that is part of the splicing machinery that will be controlled by controlling splicing. But you will see, we have a gene, SMN1, that when it is mutated, what happens is this protein is not properly made. But humans, we have a paralog gene called SMN2 in another part of the same chromosome. That the only difference with SMN1 is that it has 11 nucleotide changes in its sequence. It's a paralog. But these changes make that an inhibitor that binds to the intron 7, which is downstream of exon 7, is repressing exon 7 inclusion. So the fact is that alternative splicing of exon 7 would lead mostly to skipping, and it would lead to a truncated protein. So in the absence of SMN, this gene cannot compensate, because 80% of the protein that it makes is not good. Okay? I stress here that this is an advantage, but also it's not good. Because if we didn't have the paralog, the absence of the expression of SMN1 gene, it would be embryonic lethal, so we wouldn't see the disease. we see the disease in humans just because there is a paralog that makes little protein. This little protein is enough to survive, but not enough to breathe, not enough to have muscle movement, okay? So the idea of Adrian Krainer was not to replace or revive the mutation in the SMN1 gene, but to revive the expression of the paralog by getting rid of the inhibitor and then converting the paralog in a healthy gene to produce enough amount of protein. You follow me? Okay. So he was successful. This is Adrian. Adrian Krainer, Adrian in English, Krainer in Krainer, is from Uruguay. But nobody knows because he has no accent in English, okay? But he's more Uruguayan than the mate, and the mate is not exclusively Argentinian. When we communicate with Adrian, we communicate in Rio de la Plata, in Porteño, in Montevideano. He's very, very Uruguayan, and he's great. He got the Breakthrough Prize this year for this discovery and use. And what he found is a little oligonucleotide that now is sold with a trade name of Spinraza. And this oligonucleotide, Spinraza, base pairs with the RNA just at the target site of the negative protein. So it displaces the protein. It displaces the protein in a way that now splicing is derived to higher exon inclusion. Although SMN1 still mutated and doesn't make right amounts of protein, SMN2 now makes 80% of the healthy protein. And this is incredibly good for the patients. And Spinraza was approved by the FDA in Christmas of 2016. And since then, there are hundreds of children, you will see one, that not only survived, that can even overcome the most devastating disease. And this is the first neurodegenerative disease that has been cured in the world, okay? So I will tell the case of Cameron. I don't know Cameron, of course, it's Adrian's patient, because it's very spectacular. Cameron was diagnosed with SMA type 1, the most severe disease, when he was 5 weeks old. Doctors said he would only had 6 to 12 months to live. He could live longer, but prostrated with mechanical ventilation for life. His parents enrolled him in an experimental drug trial called Nasinersen or Spinraza by Ionis Pharmaceuticals, developed by Adrian Krainer at Cold Spring Harbor, New York. And this video shows what happened. Okay, now he's 7 weeks old when the treatment started. The treatment is intrathecal injections of Spinraza in the spinal fluid. Okay, now he's 4.5 months old. He survived, and he's grabbing objects. Now he's 1 year old. He's already holding a heavier toy. He's 20 months old. He's standing. And he was 3 years old, now he's 5 years old. He's riding a tricycle. I go to the meetings of Cure SMA, and you see the 17, 18-year-old type 1 disease ventilated with a mechanical ventilator, prostrated on a bed. And this is spectacular. Adrian is like God. When he goes to the meetings, the families go and pray. We will talk about money later. So this is an Argentinian patient, Antonella, 5 years old. She was not type 1, she was type 2, the less severe disease, one of the less severe diseases. But she was in a wheelchair, but she couldn't write properly her name, Anto. and this is before Spinraza, this is after 2 applications of Spinraza. She improved the motricity. And I talked to the parents, and there are lots of funny histories about how the patients improve, and then before the next injection, they have to, some of the symptoms go back, etc. So let me continue. So the question we had is SMN2 exon 7 splicing affected by RNA pol 2 elongation? And the answer is yes. If we transcribe the gene with the slow pol 2, you remember the slow pol 2 mutant, we have exon skipping. So this gene, or this exon, belongs to the set, the 20% family of genes in which exon inclusion goes down with slow elongation. So we reason that we have to do exactly the opposite to increase elongation. So this is the case. I'm repeating the cartoon, the movie. If elongation is fast, in this case, we will have more inclusion. So we reason that chromatin opening should increase SMN2 exon inclusion by promoting intragenic pol 2 elongation. So we decided to see what happened if we use histone deacetylase inhibitors, first in cells in culture, then in patient cells, and finally in mice. We haven't applied it to humans, but the fact is TSA cannot be applied in humans because it's very toxic, but there are other histone deacetylase inhibitors that are approved in humans, and we think that at the end we will combine the 2 therapies. But see what happens. This is exon inclusion of exon 7. When we treat with the allele-specific oligonucleotide with Spinraza alone, there is an increase in exon inclusion. When we treat with TSA alone, there is an increase in exon inclusion, but we treat with the 2 reagents, the cells, there is a higher increase in exon inclusion, which is not due to higher increase in transcription because already TSA and the ASO have exactly the same effect. So, okay, this is cells in culture. We show that TSA, trichostatin A, that opens the chromatin, opens the whole chromatin. Not only the promoter, like people assume, but it opens, this is a CHIP assay. People who understand what CHIP is chromatin improvement precipitation with an antibody against K9 acetylation. But around the alternative exon is also open, which is good. It opens and allows for faster elongation, more exon inclusion. And then we have an alternative chromatin, alternative splicing model in which we want chromatin to be open to have higher exon inclusion, mainly exon 7. But then we found that the Spinraza alone had an effect on the chromatin independently of the treatment with trichostatin A. And this is reminiscent of something we had found in 2009 with Mariano Alliot in my lab, in which we had found that certain short RNA or DNA sequences can have an effect in the nucleus by acting through the Argonaute-1 protein in a transcription gene silencing manner. And this is because these short sequences can base pair with the nascent mRNA and promote K9 methylation. And this creates a roadblock to transcriptional elongation that can affect alternative splicing through the kinetic coupling model. So we found that Spinraza alone promotes K9 methylation. But you know that K9 methylation is chromatin compaction. And chromatin compaction is lower exon inclusion. So the oligo is having a dual role. On the one hand, is displacing H9RPA1, favoring exon inclusion. But on the other hand, it seems to be promoting slower elongation, which will be, the opposite, what we don't want. So this will explain why, when we treat with trichostatin A, both mechanisms are working in the same way to promote inclusion. So we are counteracting the not desired effects of the oligo. That's why we have more inclusion and more effects. But this is cells in culture. Let me just show you that we have preliminary experiments in mice. I'm finished with this. Luciano Marasco, who is the graduate student, I didn't tell you, when the family came to us, they wanted to support us, and I say, you don't pay me anything. We will present one graduate student to the CONICET, and you will buy me the reagents. I will order the reagents to your name of the foundation, and you pay them. I don't want to touch any money from you. These are parents. I don't want to have any suspicious situation. So the mouse model, in mice, there is only one SMN gene. So if the two alleles are mutated, this is embryonic lethal. We don't see them, the pups. So the Taiwanese researchers created a transgenic mice in which they introduced the human SMN2 gene. So when you have the two alleles mutated, these mice produce a little bit of the right protein, as in the human patients. But the fact is, it's a good model because anything that would change its splicing in the human transgene will allow the mice to survive. Because with this situation, they only survive for 5 days. So they die very early after birth. So let me just show you. Luciano injected the pups at day 1, either with the Spinraza alone, or with Spinraza and trichostatin, or with trichostatin alone. So this is a heterozygous for the mouse gene, which is normal, and these are the homozygous that were treated only with Spinraza, and this is heterozygous, and these are the homozygous that were treated with Spinraza and trichostatin A. You see that they look bigger than these 4, okay? They look bigger at the same time. This is P11, 11 days after birth. This is P42, 42 days after birth. This is a heterozygous, this is a homozygous mutated treated with Spinraza alone. This is treated with Spinraza and trichostatin A. At P11, the one treated with the Spinraza alone in suboptimal doses, of course, has difficulties to turn around. The other one treated with trichostatin and the Spinraza turn around very, very easily. And this is the body weight curve. If they are treated with TSA alone, they die. This is heterozygous body weight. This is treated with suboptimal doses of Spinraza alone, and this is treated with Spinraza and trichostatin. What you are not seeing here is how many mice survive at each point. And you will see here, this is the most spectacular result. You see that if they are treated with TSA alone, they die very early. If they are treated with suboptimal doses of Spinraza like oligonucleotide, only a 20% survives. But treated with the same amount of Spinraza only once at day 1, they 80% survive. So there is an increase of survivor and body weight when you treat both with the oligo and the chromatin opening drug, which is not toxic in the mice. So the conclusion so far, I have more interesting results, but there are 2 preliminary yet, is that Pol2 elongation regulates alternative splicing through what we call kinetic coupling. That slow elongation can promote either exon inclusion or skipping, depending on the particular alternative splicing event that is considered. We found that there is changes in chromatin, alternative chromatin that could produce changes in splicing, and we are looking at a combined therapy of spinal muscular atrophy using allele-specific oligonucleotides and chromatin opening drugs. So I just want to mention Luciano Marasco, who is a graduate student who is leading the project. We have now a new graduate student coming from Italy. And all these people who are not in the lab anymore were working on the chromatin link with splicing. But we have other subjects, as I mentioned, the effect of DNA damage on splicing. And this is one part of the lab that is keeping us very excited. We study alternative splicing in plants. We found that elongation affects alternative splicing decisions. And today, we have a molecular cell paper who will be the cover, in which we show for the first time that light and dark control alternative splicing in the plant through controlling elongation. So it's the first time we have an example of kinetic coupling that occurs in a whole organism, a plant, responding to external cues, light and dark. We had the support from HHMI for 15 years, but it was ended in 2017, unfortunately. not because of us, because of them. They decided to stop the program. But fortunately, we have now support from the Cure SMA families in the United States, together with FAME Argentina, and more recently from the Lounsbury Foundation. If you give me one more minute. The case of Adrian Krainer. Adrian Krainer was studying the basic mechanism of splicing for all his life, okay? And he ended up curing a disease, a very dramatic disease, okay? So it's the best example that without knowledge of how splicing enhancers, splicing silencers, sequences, paralogs, orthologs, etc. work, there is no possibility of translational medicine. So basic science is absolutely necessary. There is no shortcut. And this is something that the authorities do not understand. They look for the shortcuts. And there are no shortcuts. And in our case, more humbly, we also are introducing something that may help to cure the disease, but because we were studying kinetic coupling for 20 years with no application at all. But without that knowledge, it would be impossible to conceive that treating with an oligo and with a TSA would help the mice to survive and to turn around, which is a very macroscopic result. So it's a fight. It's a fight. And we lose 10 battles, and we win 1. And we lose 10, and we win 1. And we hope the people from Germany who have this incredible investment in science will convince the authorities in our countries that there is no shortcut. Thank you very much.